Welcome to AP Biology Teaching Tips number 3. I'm Joy Killow. This video is on managing lab assessment. Lab assessment can be a win-win. Good for teachers means it's realistic in time demands. Good for students means it maintains rigor throughout the assessment process. Some strategies for lab assessment include using rubrics, authentic assessment, writing within the lab assessment, and creation of group posters or other presentations. I will talk about each one of these. First, let's consider the use of rubrics for lab reports. In this simple 10-item rubric, we evaluate the introductory photosynthesis leaf disc lab to be sure students understand the main concepts. After going over the rubric with the class, each student can evaluate their own paper. As they assess their paper, have them highlight each rubric item found on their paper and indicate the corresponding number from the rubric. Pause the video and evaluate this conclusion using the rubric. After you're finished, advance the video to review the rubric items that were awarded points. A similar rubric evaluation can be used for graphs. This is a simple generic graph rubric. For item 1, we are looking for a title that is descriptive. Item 2 checks that the independent and dependent variables are on the proper axes. Dry mix stands for dependent or responding variable on Y. Mix is manipulated or independent variable on X. For item 3, are the axes labeled? For 4, are the units properly represented? And 5, are the points plotted? Pause the video as you apply the rubric to the graph. When you are finished, advance the video to reveal the points. For training, you can use a generic rubric. If you provide this to students, they have a guide or scaffold for their writing. This rubric is for the discussion or conclusion part of a lab. Students use this rubric in a "Did you" mode for self-evaluation. Taking item 1 as an example, the student reflects on their writing, asking themselves, "Did you describe your questions and tell why they are important?" Pause the video to read over this generic rubric. Authentic assessment is a powerful tool. When I added authentic assessment to every lab, the student attentiveness increased dramatically. Since each student was held individually accountable, they all took appropriate interest during lab. This assessment is for the lab on osmosis and water potential and can be used formatively or summatively. I like to leave a note at the station requiring items to be put back exactly as the student found them. This leaves this lab reset for the next student. In this assessment, the students are given a potato core to mass and then several questions to answer. If you have enough time, you can do a longer lab practical. In this case, students are given 3 solutions of varying molarities, and their task is to put them in order of relative molarity. You could have multiple sets around the room with different colors representing different molarities. Not all assessments need to be individual. This is a nice group assessment. Still on the osmosis and diffusion lab. This assessment can be a wet lab or an assessment using the provided pictures. In this case, the mass is given in several pictures and the time is given. As you can see, the mass of the dialysis bag is lower after 20 minutes. The question for the students is, where is the hypertonic solution in the bag or in the beaker? Students need to realize that the bag has lost mass, and loss of mass means water moved from the bag to the beaker. This would happen if the solution in the beaker was hypertonic to the solution in the bag. To assess an electrophoresis lab, simulate a gel on paper and put it in one of your gel chambers. Then ask the question, is this gel in the chamber correctly? Look to the right. The positive and negative ends where the power supply is connected are indicated. Pause the video and answer this yourself. Then advance the video to reveal the answer. In this case, the gel is not correctly oriented. DNA is negatively charged, and it runs towards the positive pole. This picture shows the opposite. For assessment of a pGlo lab, you can provide simulated colonies in labeled petri dishes as you see here. This is a representation of a petri dish containing the pGlo plasmid and ampicillin. The students answer the 3 questions below. Pause the video as you answer them yourself. The colonies would not glow because this is an ampicillin-only plate. There is no arabinose. 7 bacteria were transformed and grew into the 7 visible colonies in the simulated petri dish. Students cannot write enough. In this example, provide this lab setup and ask the following questions to assess student understanding of both macromolecules and diffusion. This would be a good place to develop a rubric for evaluating their writing. Group posters and presentations are a meaningful way for students to reflect on their lab experiences. Post lab write-ups around the room and try a gallery walk or a post-it note critique session where students add post-it notes with comments on other students' reports. It's a good idea to mix these up, but my favorite is the whole group presentation to the class and the follow-up question and answer session. I hope this video gives you ideas for lab assessment and that will help you and your students. Authentic assessment, writing, rubric use, and group presentation are just some of the strategies that can improve student performance. Until next time, this is Teaching Tips in AP Biology.